RNA can fold on itself and make up those secondary structures. So whenever it folds on itself and makes the secondary structure, it changes its properties. So we see that in RNA, the bases are there, and some of the bases they can pair up with one another. And this pairing is actually the basis for determining the secondary structure. So while keeping in our mind, we can design some algorithms which we can come up with some optimal structures. We see that in nature, this RNA is produced as a single molecule, and those strands then they fold on themselves, and in this way then they make up those pairs and they make up those secondary structures. But here this RNA secondary structure analysis is slightly different with the DNA alignments. Over there we were looking for the matches, but here we are looking for the base pairing. So G and C, if they are present, they can pair up with one another. So we will have more score if the pairs are such that they can make some bonding with one another. So here we are not looking for the alignments, but we are looking for their pairing patterns. In nature, those structures are conserved more as compared to the sequences. So there is a concept called as covariation. So for example, if you look into these structures, so we have C, G and A and G, C and U. So in this way, it's kind of a stem-like structure. And then we observed some loop-like thing where we might see some repulsions, or where we do have the bases which do not align with one another. So that's why this loop comes, and overall structure is a hairpin. So now nature wants to conserve this structure, or this hairpin structure. So what happens over there is that when one of the nucleotide changes, for example this G changes to A in the bottom strand, at the same time the position with which it is pairing or binding, that also changes. So that kind of variation is called as a covariation. There are different arrangements with which we can represent these secondary structures. So we can have two-dimensional shapes. We can represent them with circular plot, where we can connect those bases with one another. You see there are a large number of arcs going here and there, and these arcs are not crossing with one another. So means that this sequence is actually such that it's creating a stable structure. If those arcs they are crossing with one another, so we might end up with a structure which is not, we can say, favorable, which is not stable structure. We can represent these secondary structures with a dot plot arrangement. We can have dot plots, so wherever we get the pairing patterns, we can take the same sequence on x-axis and same on y-axis. You have done it some sections earlier. So where we get a pairing pattern, we can put some mark, and while looking into the pattern of those marks, we can derive the idea whether there is pairing or there is no pairing, and what are the local region where we can see those pairings. There is some special plots, we call them as mountain plots. We can put them into a parenthesis like thing. Here we see those parentheses, so it's kind of, the structures are nested with one another, so it's showing the pattern. On one side we have right and left parentheses, they match with one another. So over there we are kind of getting the pairs which are inside, and then we can get some outer pairs and further outer pairs. So in this way we can use these parentheses. We can present these structures in the shape of trees also. Here in this diagram we can see different secondary structure shapes or patterns. For example, we can see a hairpin, so where we can see loop-like thing which is coming, so it's a hairpin loop. We can have a junction where we can see multi-loops, so they make up junctions. We see this structure in the middle. Bulges is where we can see on one side there was pairing, but on the other side there was some nucleotides which were not paired, so they move out as a shape of a bulge. So that is on one of the strands actually. We see the single strand where it starts, so we call it as single strand extensions. So actually this RNA is starting from this side and it goes all the way and wraps around, and then finally it comes somewhere here in this region. So that is one terminal, and this one is obviously the other terminal. We see interior loops present in the middle. So we have actually bulging on both sides. So bulges on both sides, they make up this interior loop. We see a stem-like structure where we see those complementarity of the bases pairs them up. Sometimes those loops, they start interacting with one another. So these structures are what we call them as pseudoknots. Pseudoknots are actually, they create instability to the structure. So normally they are not wanted as far as the secondary structure is concerned. And most of the RNA prediction algorithms, they lack the ability to predict those pseudoknots. Sometimes these pseudoknots and related structures where we can have two loops which are kind of, joined together, so they make different structures. We call them as pseudoknots, kissing hairpins and hair bulge interactions. In that case, it might go into some tertiary structures. Pseudoknots break up our dynamic programming matrices, so they create issues, so they should be dealt carefully. And as I said earlier, that is actually a good test on the reliability of the algorithms to predict these pseudoknots. One more thing is that must be checked is that the base is not already paired. So if it's already part of the pairs, this might break down the recurrence relationship. So these pseudoknots have been always the issues with these algorithms. Here we see the pseudoknots, so we see those arcs, but same way we see that some crossing edge, which is a red color edge. We can have this sequence. We can see the top part where we have labeled it as BX and BY. So this part is actually, it's where we have this top part, and this was actually the bottom part of this sequence right here. So we see the pseudoknot up there in the top part, and here we also see that this sequence starts with a base which we call it as B1, and it goes all the way and comes and ends at Bn. That is also an important trial, or you can say the test, in order to make this circular arrangement, how you would arrange those start and end points. So we see that some forms of RNA, they can form secondary structures by pairing with themselves or by folding on themselves.